Hi, my name is Lily. I'm going to be talking to you guys about the evolution of our non-coding DNA and how it connects to aging of the human brain. And so results of the study give us further insight into our genetics and can even help us find underlying causes of neurodegenerative diseases such as Alzheimer's disease. So just a brief overview of what I'm going to be discussing. I'm going to go over some background on non-coding DNA, my research questions, the results, and then the further implications of this project. For those of you who don't have a background in genomics, this is a broad overview of the role of non-coding DNA in our genome. And so as you can see, only about 1.5% of our genome is coding and actually codes for those proteins. This is typically what we think of when we think of DNA, but in reality, most of it is actually non-coding. You might remember learning in high school that our non-coding DNA is junk DNA and is not functional. But we're learning now that's not the case at all, which makes it really exciting to go into as a career. So we know that some of our non-coding DNA pads the ends of telomeres, some is spliced out of our genes as introns, some codes for tRNA. So a lot of it we don't know at all, as you can see by this question mark at the bottom. But the scope of this project is going to look at cis regulatory elements. Examples of this include promoters and enhancers. And so these cis regulatory elements, which I'm going to call CREs for the rest of the presentation, regulate the rate of transcription of the coding genes. And so promoters and enhancers are two of the most typical examples that you'll hear of. Enhancers regulate genes from pretty much any distance on the strand of the DNA, while promoters are usually found right before the gene. So a transcription factor or another activator will come in and bind to one of these CREs, and this binding will trigger transcription of the genes. It's pretty rare that it's paired one CRE to one gene. It's a very complex web that we are just starting to figure out, but we do have a lot of it documented in terms of what CREs regulate which genes. So my project and research lab is specifically focused on how these CREs have evolved over time and which ones are functional and unique in our human brain. And so a previous project in the lab found answers to these first questions, and I will be using those answers to answer this last question: which of these CREs regulate important aging processes? So if we can answer these questions, we'll have a much better understanding of how human genetics works, and knowledge of this can be used to develop medical treatments and further understand human aging-related diseases. So this is an overview of the data set that I started off with. Using statistical techniques, they found 722 CREs that are both evolutionarily conserved among vertebrates. So this assumes that they're functional and doing something. And then also experience significant positive selection in humans when compared to other primates like chimpanzees. And then they also found 179 differentially expressed CREs that have high brain activity, using an assay called Lenti-MPRA. And of these CREs, only 34 overlap between these two categories. And so I'm looking at, of these CREs, do any of them regulate known human aging genes? So if we can answer this, we can understand which unique and brain-related CREs are regulating some of human unique aging phenotypes that other primates don't necessarily experience. So no need to read and memorize all of these genes, but these are the 64 aging genes that I picked out based on literature review. Many of them function in longevity, but some of them are a bit different. Some code for telomeres, and some are even tied to dementia or other cognitive disorders like that. And so then using genome databases and RStudio, I was able to find which of these aging genes are regulated by any of the CREs that were described in the previous slide. And so of the 64 aging genes that I looked at, I found 6 were regulated by those CREs that I discussed. And so 3 were regulated by CREs with high brain activity. So that's AKT3, CDH13, and WOX. And so I don't find it very shocking that they're regulated by CREs with high brain activity just because their functions imply that they're active in the brain. but I was really excited to see these genes that are regulated by CREs that are selected for evolutionarily, particularly CDKAL1, which actually is regulated by a CRE with both positive selection and differential expression. And so these make these three genes very promising candidates for looking at how regulation of them has changed in relation to human aging compared to primates such as chimpanzees. And so this figure shows the distance between these aging genes and their CREs using data from the UCSC genome browser. And the reason there's more than 6 counts on that left side is because each CRE and each gene often span multiple different regions. And so what this tells us is that the aging genes are most often regulated by CREs that are closer to them rather than farther, which implies that a lot of these CREs might be promoters rather than enhancers. However, the distribution isn't extreme, so regulation is still coming from CREs that are up to 2 million base pairs or nucleotides away from the gene. And so I was also curious if those 6 genes are actually found in our closest ancestors, and I found that they all were almost exactly in terms of how many amino acids. And so this is just further evidence that what's changed evolutionarily is probably the regulation of these genes and how often they're produced and expressed, rather than the existence of the genes themselves. So in summary, I've been able to identify specific CREs that regulate our aging-related genes that are either evolutionarily selected for or have high brain activity, or even both. And so this gives us an in-depth understanding of what their causal role is in our human neural phenotypes. And more broadly, we can take another step towards understanding what genomically makes us human, and how our brains develop and age over our lives. So it's important to note that these categories of selected CREs is based on our own parameters. So if you use slightly different parameters, we may identify different CREs. And so this is why more studies definitely need to be conducted in order to verify findings and investigate other CREs and other genes. Finally, aging is a huge risk factor for the development of neurodegenerative disorders such as dementia or Alzheimer's. So in the future, these genes in their regulatory regions should definitely be further investigated when looking at possible genetic drivers for these diseases. So that wraps it up. Thank you so much for listening. And if you have any questions, please let me know. Thank you.